back to Paleo Talks. Good to see everybody out there. Everybody chiming in. Keep chiming in with where you're from. Happy to see you. We are on episode 55. Hard to believe it. And today, special guest, Aly Baumgartner. How you doing, Aly?
Oh, I'm good. I'm just burning up here in I'm in Kansas now. I was going to say Texas. I don't remember where I am.
Well, wonderful to have you. We'll get into your background and all of that introduction in just a minute. But just to let everybody know, Aly used to be here at ETSU. So great to see you after the PhD and with a job. And again, with us here, David and Chris. How are you both doing?
Good.
Very good. On Proboscidean steppe somewhere.
Yeah, I needed something a little chilly.
Well, let's jump over to David and he can remind everybody how the show works throughout.
Sure. Welcome again to Paleo Talks. We're getting a lot of comments in the comments, as we like to see. This Paleo Talks is going to be the same as our typical format. In a minute, we're going to introduce our guest more in depth and then go into our main presentation. During the presentation, please think of questions that you might want to ask Aly today. And then once the presentation is over, we will open it up. The rest of the program is going to be Q&A. So at that point, we'll remind you to leave your comments in the Facebook chat comments section. And as always, if for any reason you can't leave a comment on Facebook, I'll be keeping an eye on the Gray Fossil Site accounts on Instagram and Twitter, so you can leave us your questions there.
All right, thank you, David. Just to remind everybody, we're coming to you from the Center of Excellence in Paleontology at East Tennessee State University, where we oversee the research on the Gray Fossil Site and other localities. And we have this extraordinary paleontology program and fossil site just right underneath us and outside the door here. And today, again, we have Aly Baumgartner. It's so cool to see you. It's amazing to see our alumni.
Thank you. This is fantastic.
And I don't know if you've watched many of these shows yet or not. Maybe you haven't seen any of them. You don't have any idea how they work or what we're going to ask you. But the first thing we always ask is, tell us about your background, why you got into paleontology in the first place, and that whole story of what got you interested as a child, maybe, and
all right.
through ETSU, and now where you're sitting out in Kansas.
All right, cool. So I am currently a paleobotanist, sort of. I got lost on the way. So I grew up in Michigan, and one of the things about living in Michigan is that everything around you is some sort of glacial landform. So I thought that was the coolest thing. The fact that you could so easily, like constantly farmers were digging up just casual mastodon bones, like that was amazing. The fact that like my grandparents' gravel driveway was made of fossil coral, I was just utterly fascinated. There was never really a time in my life when I didn't want to be a paleontologist. And so I was not a dinosaur kid. I was a mammoth kid, like Ice Age megafauna, my jam.
Yeah.
So I went to the University of Michigan for my undergrad, and while I was there, I worked in the fossil prep lab. I was a tour guide. Fantastic. Like I was convinced that I was going to work on proboscideans. I did two internships at the Mammoth site in Hot Springs, South Dakota. I was pro mammoth all the way. And then I applied at ETSU. I got into the biology program. And when I met my advisor, he was like, okay, so you're going to be studying fossil plants. And I was like, oh, I am. Okay. Because I had taken a bunch of botany classes, but it was because I was interested in like Paleo Environment. And so I was like, okay, plants. Sure, I'll try that. And realized that, just kidding, I loved plants. Like that was really what I was most interested in. And so
now, I have to stop you for just a minute because this is fascinating. So when you came to ETSU and you knew that there was a vertebrate program.
Well, no. So maybe that's the thing. I didn't know. When I applied, y'all had just split.
Yes.
It was only within a few years that paleo had gone from bio to geo. So when I applied, someone in the biology department called me, and they were like, are you sure you want to do biology or do you want to do geology? And I had just gotten rejected from South Dakota School of Mines for not having enough of geology background. And so I was like, oh, no, bio, definitely bio. And then when I got there, that was when I realized like, oh, wait, all the geology, like all of the paleontology I want to do is over the other department, which is, so my first year I took all these botany classes and I really enjoyed it. It was what I was very much interested in, but it was my second year. So the summer in between, I started volunteering at the Gray Fossil Site. And then my second year, I took paleoherpetology and all of those classes. And that was fantastic. And that has been so useful in my job, just saying, having taken like herpetology classes, because it helps with mosasaur anatomy, just saying. But anyway, after my master's, I did an internship at Florissant Fossil Beds, working on fossil plants. Absolutely loved it. Fantastic fossil site. I actually worked as a substitute teacher after that, which was very fun because I got to be like a visiting scientist of like your friendly local paleontologist. And then, as I was saying a little bit before we started, I actually met my dissertation advisor on Twitter because I decided, like, you know what, plants are really what I'm most interested in. I want to do a PhD studying fossil plants. And so I contacted Dan. Actually, I contacted a couple people. I first talked to Peter Wilf at Penn State. And when I talked to him about what I was interested in, he's like, you should really talk to Dan Pepe instead. I'm like, don't worry, he's on my list. And so I did my dissertation with Dan at Baylor. I was there for 5 years. And I did my research in Kenya. I did a few months of field work in Kenya, which I'll be talking about today. But I was set to finish up. I was supposed to defend sometime in March of 2020. That didn't happen. I had my first job interview for this job the day before Baylor shut down in March of 2020. So I was like, cool, not getting a job. I've accepted this. This is fine. And then 3 weeks later, I got a second interview, and then I got a job at the Sternberg Museum at Fort Hays State University as the paleontology collections manager. So I defended my dissertation on July 2nd. That was a Thursday. I took the Friday off. I spent all of Saturday and Sunday packing. And then I got the U-Haul on Monday and left on Tuesday and started my job the next week. Like, there was no time in between. Yeah, and so as the paleontology collections manager here, I am in charge of the vertebrate and invertebrate paleontology collections, as well as the geology collection. There is a paleobotany collection, but I am not in charge of it.
And you said nobody is.
Nobody is, unfortunately. Yeah. So if someone wants to give us a whole bunch of money to take care of that, that would be great.
This is a good place to advertise for that. David often gives out a lot of money, so.
Yes. Yes, please give us money.
All right, well, thank you for that background. That was fun. And what we'll do now is we'll go ahead and move forward and start with your presentation, if you could share that.
All righty. All right. We got to do the thing where I figure out how to share my screen. As a collections manager, I don't often have to share my screen because I don't teach. All right, so let's see here.
Just move around your cursor. You should see a share screen green in green.
Oh no, is it not going to let me? Oh no.
All right, we'll work through these technical issues.
Okay, open system preferences. I might have to leave you and come back. That'll be super fun. We'll find out. I hope
we will talk about what we know about plants, which should fill the entire 5 minutes it takes.
David, you know things about plants.
I do. I know things about specifically like 6 topics in plants.
Well, the only advice I have is moving around your cursor till you see share screen. Does anybody else have any other advice?
Do you see the share screen option on the bottom?
Oh yeah, no, I can do it, but when I go there, it gives me like a danger button, like, oh, you're not allowed to do that. So I need to update my system preferences on my computer.
Huh. Well, is your presentation a file that you can send?
Yes. So we can do that if you want. Yeah, we can do that. Otherwise, I'm going to need to quit Zoom, and I don't want to do that.
Cool. Yeah, if you email it to me, I can do that. I can be the one who's in charge of it, and you just tell me when to go to the next one.
I can tell you what to do. Delightful human.
That's right.
David, have you emailed me on this? Oh my gosh. David, you've not emailed me from this.
I will send you
This is the first time that we haven't asked the presenter
in
to.
the chat.
We were so distracted
So,
because we all
yeah.
know each other.
Yeah, we were.
That was the
We
that was
used
the
to do
problem.
that at 11:45.
But we were having so much fun just catching up. I haven't seen y'all in so long.
Mm-mm.
All right, cool.
We'll get this working. Yeah, once you send that to me, I can open it up
Okay.
and then I can
Yeah.
share it.
Okay, if my computer will decide that this is allowed. Hi guys, this is great. Fossil plants are fantastic.
It's funny to hear that background because it's exciting for me to see Aly join the Paleo Talks today because Aly and I overlapped in our time at ETSU. That's how I know Aly is through when we were both students and we would work out at the Gray Fossil site together. And it's also funny that you mentioned Peter Wolf because Peter Wolf was technically my advisor for a little bit when I was an undergraduate, when I was at Penn State. So as always, paleo is a very small world. Yes.
I will have to rejoin Zoom because my file is too big because it is full of pictures. So
Gotcha. All right.
No
all right,
problem.
one
right
second.
ahead.
All right. I need to, I will be back. Be kind to each other while I'm gone.
Okay.
Will do.
All right.
All right. Thanks everyone for sticking with us. Sorry for the technical difficulties today. Let's see who
We can blame it on Kansas, I think.
Yeah.
So we've got a Chris watching from Hayes. We've got Susan from California. Greg McDonald's here, reminding us that while at Baylor, Aly did work at the Waco Mammoth site. Very cool. We'll be back on here in just a moment. This will be fun because we don't get to do a lot of plant discussion, and it's a common point of discussion among paleontology that there are certain topics in paleontology that get a lot more attention than others, and big flashy vertebrates like the three of us tend to overshadow plants sometimes. So it's nice to be able to highlight the greener side of things.
But they're some of the most exquisite fossils. I mean, and seeing a leaf impression is, I mean, that takes your breath away. Sometimes like a mastodon femur. Well, I will say that even as a mastodon guy.
Well, and plants are also make up some of the most famous fossils. You know, even people who haven't heard of a lot of fossils have heard of like petrified wood.
Oh yeah.
You know,
right.
Plants are very common. They're really common portions of the ecosystem. Greg McDonald says herbivore workers need to think about plants. That's true, Chris.
Yeah. Think about plants all the time. Yeah.
Yeah. It's funny how much plants come up in discussions at the Gray Fossil site, even though we don't on site do a lot of plant research.
The amount of time I've spent with my head buried in pollen reports is really odd for a person who does vertebrate paleontology.
Well, it is amazing, everybody, that we're on episode 55 before this happened. So,
Oh yeah, we've got a pretty
before we
good track
actually
record.
had that many without technical difficulties.
One of the
I think
things
it's my background. It must be
It's
my
your background, Chris.
Yeah.
We've had enough with proboscideans. That's it.
It's been exciting the last couple of years at Gray. There has been a lot of news with plants. We just recently had a publication of a new species of passion flower at the Gray Fossil site, which is really cool. That's one of Tennessee's state flowers.
It's something that's still around. People can see a passion flower today and kind of make that connection. So it's been around a long time.
All right.
Hey, Aly.
Let's try again.
All right.
Ah ha ha ha. Bueno. Love it.
All
Well,
right.
good. We hit the end of everything we did.
That was
We
it.
have-
That was. I was
we
just
have-
going to have to start playing podcast episodes.
Of me talking because we could do that.
We
I was
could.
gonna
Okay, hold
show
on a
some
second.
plants from my tea bag I have over here.
Okay, hold on. I gotta switch screens. All right, cool. Wonderful. Yeah, David, you should know things. Like you could do this. You don't need me.
I recently got a message from a friend of mine who was having an argument with her fiance about what biome East Tennessee technically counts as. And I said, hang on a second. I asked this question on a podcast and I can play you the answer. So you were very
really?
helpful. Yep.
All right. Can you see my screen?
It looks great.
Wonderful. All right. Let's see if I can make the slideshow go. Please. All right. You see the wrong screen? Let's swap displays.
Yep.
Now you see the right one, correct?
Perfect. Perfect.
Bueno. Thank you for your
Fantastic.
patience.
We did it. Thank you, everybody, for sticking with us. We've still got our crowd here. People are still excited to see.
Good,
let's go ahead and
fantastic.
get started.
I will make up for my inability to prepare with vast amounts of enthusiasm. So as we have said many times, my name is Aly Baumgartner. I am the Paleontology Collections Manager here at the Sternberg Museum of Natural History. And I'm going to talk to you about why plants make the best time machines. So starting at the very beginning, plants are, in fact, the best time machines. They are non-modal, which means that plants don't move, generally speaking. So their options, if the climate or the environment changes, is basically to adapt or die. And that means that plants make fantastic local indicators for paleo environment and paleoclimate, because if they can't survive, they can't just like wander off. They are also, there we go. Okay. They have fantastic preservation potential because there are just so many parts of them that can be preserved. And unlike most animals, they can just willy-nilly lose bits of themselves without dying. So losing branches or leaves or fruit is not a death sentence for a plant. So you have so many more opportunities to be preserved. Wood and pollen are famously common fossils, and leaves are much more common than people tend to think. And then finally, plants are photoautotrophs, which means that they eat sunlight to make energy. And that ties them intrinsically to their environment. They make their own food, so they are really limited by what the temperature, precipitation, sort of other things are. So when we talk about paleo environment, we tend to be talking about biomes. And the thing I love about biomes is that they are named for the plants that are there. So when I talk to people and say that plants are not just your green background, this is what I mean. Like, environments are plants. Animals live in environments. Plants are environments. So if you look at this Whitaker diagram, this is what we use in paleobotanical paleo environmental reconstructions. And it's basically showing the relationship between annual temperature and annual precipitation to really shape the sort of environment that you're going to have. And so, yeah, if you look at just the names of these environments, grassland, named for the grasses, woodlands, shrublands, they're named for the plants that you can find there. And if you look at the globe, that's very obvious, right? With the three main colors on this map are blue, green, and white. And two of those things are not alive. The other one's a plant. So if you look at this map, you will see that you can tell where different environments are by just looking at the plants there. You have these darker areas with taiga, boreal forests, or tropical rainforests. You can see the grasslands in stark contrast to the desert. So just by looking at the map, what we're really looking at here are plants. And there is more to plants than just leaves, but I personally focus on leaves, and leaves are very useful in the fossil record. If you've ever talked to a botanist, and I really hope you have, if not, you can talk to me. I'm very nice. If you've ever talked to a botanist, they will tell you that leaves are the worst way to identify a plant. They can be so variable. Oak trees, for example, are so variable in their leaf shape. The sun leaves at the top versus the shade leaves at the bottom are going to look really, really different within a single plant. But that plasticity in shape is what makes them fantastic for estimating paleoclimate. So if you look at these two leaves, on the right, you will see that leaf is a lighter pink color. That is an indication that this leaf is probably a little bit thinner. You tend to have thinner leaves when you have deciduous leaves, because if you're just going to drop your leaves seasonally, there's no point in really investing a lot of resource into them. You'll also notice that the edges of the leaves are crinkly, they're kind of toothed. And you generally tend to see more taxa with toothed leaves in places with cooler mean annual temperature. So just by looking at this leaf, we have a hint that maybe it came from somewhere a little bit cooler, and someplace that would have some degrees of seasonality that would make the plant lose its leaves. If you look at the leaf on the left, you'll see something a little bit different, right? It's a darker pink color. This is probably a much thicker leaf. You tend to see thicker leaves when you're looking at evergreen leaves. So think of like magnolia leaves. Those things are like leather. If you look at the edges of the leaves, you'll see that they're very smooth, and you tend to see a higher proportion of entire margined or smooth margin leaves in places with a higher mean annual temperature. So what we're looking at here is a leaf that is probably from some place that is a little bit warmer, less seasonal. But again, we're looking at 2 leaves. You can't really say much from that. But leaves are absolutely amazing in that they have what's called phenotypic plasticity. So that means that the phenotype can be variable. So genotype is literally what the proteins code for. It's literally what is in your DNA. Phenotype is the kind of wiggle room in the way that is actually expressed. So your genes may say you have green eyes, but your phenotype is like, how green are they? We see the same thing with leaves. So if you look at this figure, you have 3 rows of leaves. These are all from the same species. These are all from red maple leaves that were grown anywhere between Canada and Florida. And by looking at them, just generally speaking, they all look pretty similar, right? They're all toothed and they're all lobed. But if you look at the leaves in the top row, they're more jagged, they're toothier, they're lobier. But if you look at the ones at the bottom, they're a little bit smoother. The edges have been smoothed out a little bit. And that makes sense given changes in temperature. So the leaves on the top are from the coolest areas, the leaves are on the bottom are in the warmer areas. So this phenotypic plasticity, this relationship with climate and leaf margin happens on different scales. It can happen on species scale, but it can happen within a species across its range. And this is fantastic because it means that we can use this knowledge to reconstruct paleoclimate. And this is something that paleobotanists have known for more than 100 years, this relationship between leaf margin and mean annual temperature. And so you will have univariate plots like this where you take one variable and get out another one. So you take the proportion of leaves, a proportion of species in a flora without teeth against temperature. And as you have fewer things with teeth, you tend to have higher temperatures. We'll come back to this because it's slightly more complicated than it seems at first. So in addition to being able to look at the morphology, the shape of the margin to get at paleo temperature, we can also look at the size of the leaves to estimate precipitation. So plants are photoautotrophs. They photosynthesize. They use their leaves to collect sunlight and bring in CO2. So CO2 is collected with teeny tiny pores called stomata in the plant. However, that can allow CO2 to come in, but it also allows water to escape. And water is a smaller molecule than CO2. So water can escape faster than the CO2 comes in. So it is a constant battle for the plants to have leaves as big as you can in order to photosynthesize and maximize that without losing all your water to the environment. So there is a relationship. The bigger the leaf, the wetter the environment. So if you look at the leaf on the left that's getting a nice hug from David Attenborough, you can see that that is a massive leaf. That is probably a very wet environment. Versus the euphorbia on the right, I took this picture in Nairobi National Park in Kenya. And that is, euphorbia are similar to cacti. They have basically the same adaptation to deal with water loss. They got rid of their leaves, turned them into spines, and made their whole body photosynthetic. So just by looking at that, we can tell that this is probably a much drier environment. And again, we got a graph for that. So there is a relationship between leaf size and precipitation. It's not quite as beautiful and direct as temperature. This is in log scale, but we see the same general trend that you tend to have much smaller leaves in drier environments and much larger leaves in warmer environments. But it's not that simple. So one of the things that I looked at during my dissertation and that I'm very interested in looking into is the regional variation, where a plant is growing and where its lineage is from affects this relationship. So if you look at these two plots, these are the graphs that I showed you before, but they are color coded based on whether or not this is a Northern Hemisphere or Laurasian, so Northern Hemisphere minus India, or Gondwanan lineage. So Gondwana is the Southern Hemisphere plus India. And in general, they actually seem to have different relationships. So if you look at the graph on the left for temperature, you'll see that that black regression line is if you take the whole data set. But if you look at the pink dots, that's just Laurasia, so Northern Hemisphere minus India. And that's actually not that different from the general regression line. But if you look at the blue diamonds, that's Gondwana. That's a fundamentally different relationship. So that's something that needs to be explored. Similarly, when we look at precipitation, we see that generally speaking, all of the Laurasian sites tend to cluster together, and we have a little bit more variability in the Gondwanan floras with that relationship between leaf size and precipitation. And that makes sense. That's something that we should really explore, because if you just look at the map, the Northern Hemisphere and the Southern Hemisphere they're fundamentally different places. Even just broadly speaking, the orientation of the continents, you tend to have much wider continents in the Northern Hemisphere and much narrower continents in the Southern Hemisphere, meaning that you have these great continental interiors that aren't as prevalent in the Southern Hemisphere. And the other thing too is that your models are only as good as the data that go into them. So this is the calibration data set for a multivariate model that is called DILP, digital leaf physiognomy, that is very common in paleoclimate reconstructions. And as you can see on that graph on the right, graph on the left, that those are the calibration data sets. Those are the places where the data was taken from. And you'll notice that Africa is so not important to this model, it got split in half in the map. So one of the things I wanted to see, given that I was studying a Kenyan fossil site, was what happened if you actually added data from Africa. So I added, I think it's 18 different modern floral sites from modern Africa that were a variety of different temperatures, similar temperatures, variety of precipitation. So I really wanted to fill out the tropical seasonal forest and tropical rainforest part of my map. And what I got was this. These are my favorite graphs in the whole world. They are called canonical correspondence analyses plots, but really they look like snails to me. So what this is doing is it is taking the known temperature and precipitation and plotting the leaf characters. So all of the, and then color coding it based on what type of environment it is. So in general, floras with similar characters tend to be found in places with similar temperature and similar precipitation. And so the circles are the calibration data set, and the diamonds, nope, those are triangles. Shapes are hard today. The triangles are the sites that I was trying to predict. My modern African sites, based on the data that we had, where did this predict that the modern African sites would fall? And none of them fell within the proper biome, and many of them fell outside the plot. And then if you look at the graph on the right, I tried this with two of my fossil localities, my Kenyan fossil localities, and neither one of them plotted in a regional biome, and one of them's not even on the map. And so we wanted to know why. What is it about these African floras that has a fundamentally different relationship than these temperate and northern hemisphere floras? So if you look on the right, basically The longer the line, the stronger the pull. And you'll notice that a lot of my sites are being pulled towards having a greater leaf area than you would expect for a given precipitation, and a higher percent of untoothed leaves than you would expect for a given temperature. Basically, the trends I just showed you for leaf area and toothiness don't quite work the same way in Africa. And so I was afraid, what happens if I try to calibrate my data set? What if I add the African floras and it completely ruins the model? Fortunately, it didn't. It just expanded our knowledge, our understanding of what it means to be these types of floras. So the scale is the same on these two figures, but as you can see, the snail is much bigger in the right graph than the left. And that gave me the confidence to apply this to some African floras. So this is the second paper from my dissertation that was recently published, looking at the paleoenvironmental changes in the Hawegi Formation, lower Miocene, of Rasinga Island, Lake Victoria, Kenya. So to give you a little context to orient you in time and space, I did my research on Rasinga Island in Western Kenya. It is an island in Lake Victoria. You can see it highlighted in the map there. So it is surrounded in yellow. It's not particularly big. I think it's about 3 miles, about 10 miles long and about 3 miles wide at the widest part. Not particularly big. But you'll notice that it is very close to the now extinct Kisangiri volcano. I say now extinct because 20 million years ago, in the time that I was studying, that was an active volcano. You can tell because there are volcanic layers below and directly above the localities that I was working at. The other thing to note is that there was no Lake Victoria during the early Miocene. So there was no water to protect them, but it also meant that you had influx from the mainland. So keep that in mind. Rasinga Island is basically the best fossil site in Kenya. That's my favorite. I might be making a bold statement. Most people won't agree with me. That's fine. Love Rasinga Island. So there's been fossil collection since the 1930s. Over 75,000 fossils have been collected, over 100 species of mammals. And so you have things from these really charismatic primates. It's no longer called proconsul. It's actually akembo. But you also have these scary guys, creodonts, you have hippo relatives, you have calicotheres, and you have other things like rabbits, lots of really cool invertebrates, ground burrowing bees will find their little hives. It's really cool. But what I'm most interested in is the context. When I was doing my research in Kenya, I would say I am the context. And so it's important to really understand the environment that these animals were living in if we want to know anything about early hominid evolution. And many attempts have been made to reconstruct the paleo environment at this time. So anything from a closed canopy tropical rainforest to a mixture of woodland and forest habitats to open and semi-arid environments to a mixture of open and closed habitats. Everything, literally everything, just like these are the possible environments. So that's not useful. So what we wanted to do was look at 3 floral localities within the Hawegi Formation. And it's kind of delightful how evenly spaced they are in time. So at the base of the Hawegi Formation is the Kiahara Hill flora. This has not been previously published on because there are no vertebrates known from this site. It's about 18.3 million years old. Above that is the R5 locality that has been previously published. I didn't do any additional collections there. I'm just using it as a comparison because there are vertebrates there. People took care of that one already. That's about 18.1 million years old. And then finally, at the top of the Hawegi Formation is R3. I did additional collections there. There has been extensive research done at R3. It's the coolest place ever. Except for the Great Fossil Site, of course. And it's about 17.8 million years old. So by looking at the size and shape of the leaves from these 3 sites, we were able to show that the there is some variability in the paleoclimate through time at these 3 localities. So if we're starting at the base of the Hawegi Formation, our Sumikia-Hara Hill, that is pretty solidly in this tropical rainforest to tropical seasonal forest. So this is going to be a pretty wet environment. As you move up through time, then we have R5, and that is solidly squarely woodland. This is a much drier environment, actually no difference in temperature. And then as we continue to go up in the section, continuing on through time, we get to R3, and it's kind of somewhere in the middle. A similar temperature, wetter than you see in R3, or excuse me, in R5, not as wet as we see in Kiahara Hill. So let me take you through time. In my time machine, we're going to start at the base of the Hawegi Formation at Kiahara Hill. So how did we do this? So we were looking at a whole bunch of different lines of evidence, because take everything you can get. If you have this kind of resolution, that way you can kind of refine your assumptions a little bit. So we were able to find these absolutely beautiful fossil leaves. They were very big. In the words of my colleague, Peter Wilf, you can't get big leaves out of small rocks. And we were struggling to get blocks out that were big enough to get these complete leaves. Not a single leaf that we found at this site had teeth, indicating it's probably pretty warm. We also found these extensive branch casts, meaning that you were probably having a pretty complex canopy situation. So this is the graph showing the different morphotypes, the different shapes of leaves that we found. And as you can see, none of them had had teeth. That HW59 is probably my favorite. It was absolutely huge. We never got the whole thing. We got parts of it. We got the base of one once. It reminded me of an umbrella. It was very large. One of the other things we were able to do because of the quality of preservation at the site was look at leaf mass per area. So I talked at the very beginning about leaf thickness and how that is related to leaf lifespan. So the thicker the leaf, the longer it will live. And so we compared our fossil sites to a handful of different modern African localities to just get a sense of what this would have been similar to. And the similarities are somewhere between modern tropical rainforest and modern tropical seasonal forest, which makes sense given all of the other lines of evidence. It's pretty much sticking us in the same place. So a modern analog for Kiahara Hill might have been something similar to the tropical rainforest and seasonal forest that we see in West Africa today, like this forest in Cameroon. If we move up through the Hawegi Formation, we get to R5, which is an utterly fascinating site that I did not spend very much time at. This site is giving us a variety of different indications of what this environment was like, which is utterly fascinating. If you look at the sedimentology, we have evidence both of like fluvial flow indicators, we have ripple marks, as well as these evaporitic layers showing that, yes, there was flowing water, but there were also these periodic drying events. The vertebrate fossils here are really cool. There is a crocodilian bone bed, so multiple specimens of the same species. Again, this is indicating some degree of standing water. The plants are reinforcing this. They found sedges and reeds, the kinds of things that you would find maybe next to a pond. The fascinating thing here is that we also find a few different species of primates. So we're getting things that are saying slightly more open environment generally. And so this is the artist reconstruction from this paper. I adore it. It is so extra because it's really playing into the, yes, there was water, but things died here. Like I really enjoyed the skeleton on the banks of the water. And then as we continue up to R3, this is such a cool locality because so much work has been done here from previous paleobotanical work, paleopodology, vertebrate paleontology. It's spectacular. So they've found these in situ stump casts with these branching roots. So not only do we have a good sense of the size of these trees by looking at the roots and the diameter of the trees and their proximity to other stumps, they were able to use modern forestry methods to reconstruct the canopy cover. So based on the size and the proximity of the trees, it was probably a multi-storied canopy, which makes sense when we look at the leaves. And when we look at things on the landscape, the leaves are a little bit smaller than we saw at Kiahara Hill. This is the only site that had any leaves with teeth. And yes.
So I see that you have a Calicothere there, but you were saying you don't have any fossils in this site.
Oh no, this site has fossils.
This is the one with. Aha.
This is the one with all the good stuff. So yeah, we have primates from here. We have Calicotheres from here. I can't even remember. Flying squirrels, like so many cool things. The thing about it is, okay, so brief diversion, I blame Blaine entirely for this. So the difference between vertebrate paleontology fieldwork and paleobotany fieldwork is the level of destruction. So for vertebrate paleo, and I've done both, I can confirm this. So vertebrate paleontology is a lot of prospecting, like just killing your back, staring at the ground and like poking things. Is this a bone? And then trying to figure out where it came from and like, okay, is this a layer? Versus paleobotany, we just move mountains. We, instead of, because the leaves are just going to weather out, we're not going to see them. So instead, we get good at identifying the likely fossiliferous layer in the rock, and then we just take out blocks and split them open, which means that we take out mountains, we take out hillsides. So because Kiahara Hill, where I'd worked before, there were no vertebrates known from that site. We didn't see any vertebrate material while we were there. They've prospected there periodically and not found anything. So I was given fair reign to just take out rocks.
Does that mean that you were throwing vertebrates everywhere? Or
so that's what I'm going to get to. So we checked, don't worry. But when I was doing field work here, the week before I came, they were thorough. They went through and just made sure that they weren't going to miss anything before I took out like couches worth of rocks. So don't worry, they were prepared for me. They dealt with paleobotanists before. They knew what they were getting themselves into.
Well, sometimes when we talk about a place that has vertebrates and it has plants, you might be talking about a little bit of a different layer, perhaps. But
yes,
in this case, it is in the same.
It's very close. So there's probably a little bit of time averaging, but given the like connections that we are able to find, so the leaf layers and the soil layers and the stump layers are the same, and they match what we're seeing with the animals. So, and I think it's not this site, but it is another site nearby where they did find vertebrate fossils in a stump. So,
cool.
Which is really cool. So not this one. So we're doing a little bit of connecting the dots, but like they're not that far apart. So we're pretty confident like we can probably combine this. But that is a concern because that doesn't happen very often. But it is really cool because like the way that the site is set up, we were able to do a couple of different things. So for example, they had already done they'd already looked at the stumps, they've done that. I collected leaves. And one of the things I was able to do because of the size of the locality that I was working in, you can see it in the picture of the bottom, we were able to, by marking or monitoring, I guess, how the locality of the leaf sites to the stump sites and looking at the taxa that we found there, we noticed a couple of patterns that in general, we tended to have just woody die-cut leaves, just tree leaves in the places closer to the stumps. But as you got further away from the stumps, you tended to have the tree leaves along with monocots and emergent aquatics. So things that are going to be rooting under the water and the leaves are coming up. So not specifically water lilies, but that sort of situation. And that's really interesting because there was a little bit, okay, so when they were previously working at this site, there's a little bit of ambiguity with what the paleopodology was saying, what the paleosols were saying, because like the interpretation could go one of two ways. And then once I was able to come in with the leaf information, like, hey, I'm finding emergent aquatics, that made it pretty clear that what we were seeing from the paleosols was periodic ponding, episodic ponding, seasonal ponding. So we are seeing this variability on the landscape, which is very interesting and something that, like, it takes all of these different pieces of the puzzle to put it together to get this kind of picture. And this is why one of the reasons I absolutely love this locality is there's no shade, which is no good, but other than that, everything else about it was great. There are plenty of fossil primates known from this site, and in fact, the same species are found at R3 that were found at R5, which these are very different types of environments. So wrong direction, there we go. So these are the common leaves that we found at R3. So HW23, the huge piece of the pie, that has teeth. Basically everything we found was that, which is very interesting considering the other two sites did not have anything with teeth. And HW24 has teeny tiny little teeth. So this has a higher proportion of things with teeth. Fun fact, that could be related to lower temperature, probably related to lower temperature, but in general, you tend to find a higher proportion of taxa with teeth in places with near, like riparian communities. So places with more water tend to have a higher prevalence of teeth than you would expect. So that also makes sense. If we have a site that's like, we know that there was ponding and this in the environment, it might make sense that we might have toothier leaves. And then again, when we look at the thickness of the leaves and compare it to other modern African floras, we see that again, it's kind of this in between, mostly tropical seasonal forests, but a little sprinkling of woodland. And this is the artist's reconstruction of the site. I absolutely love it. If for no other reason than they got the leaves right, they got the plants right. And that doesn't always happen in paleo art because people always care about the animals. So I'm here to speak for the trees and to get really excited about that. But to kind of sum it all up, when you're looking through time in the Hawegi Formation, you're running at the bottom, you're seeing this tropical rainforest, a tropical seasonal forest situation, moving up into a drier woodland environment, and then getting a little bit wetter into this tropical seasonal environment. The climate variability is primarily in mean annual precipitation. So that is what's leading to these environmental changes, is precipitation changes, which is very common in equatorial Africa. You don't have that much difference in temperature. It's the changes in precipitation that drive environmental change. Finally, the most exciting thing is that the overall mammal communities varied between the two sites where we were able to look at that, with the exception of the primates. We saw the same species of primates at both localities, which means, unsurprisingly, that hominids are adapted to variable environments. It also means that you cannot use the presence of primates as an environmental indicator because, like, y'all, we're adaptable. We always have been. Like, we can deal with anything. So thank you. I had so much help when I was in Kenya and in many institutions around the world. I had an army of undergraduates who helped me process leaves. I appreciate them so much. And yes, here's a picture of my destruction at R3. And the way that we removed the leaves from the locality was we would wrap them in toilet paper and then tape them up with masking tape and load them full in basins and then walk them to the Land Rover. It was fantastic. So thank you.
All right, thank you, Aly. Just fascinating. Just a question about the field work itself. Tell us a little bit more. What was it like? Where did the fossils go when you were done?
I love this question. All right, cool. So the vast majority of the time that I spent in Kenya was not doing field work. So I spent a month, then 2 months, and then a month in Kenya total. So vast majority of the time I was actually at the National Museums of Kenya in Nairobi. So all of the specimens ended up there. And that's great. I'm more than doubled the paleobotany collections at the NMK with all of the stuff that I brought back from Rasinga. It was fantastic. I have a picture. I should have included it in here. I personally filled the Land Rover with fossils. The way that we would do it is we would wrap them up, and then we would buy these trunks on the island. And I don't even know, 6-7 trunks I filled full of fossils, and they're rocks. So they're very heavy. But yeah, so all of the fossils are at the NMK, which is fantastic. I spent a lot of time there. I love the humans there. I love the fossils there, but I love the humans there. But field work in Kenya was some of the most fun I've had. So getting out there was an adventure. I was joining a field school, and I was meeting them on the island, and I was originally supposed to fly to a nearby town and someone was going to pick me up. But that airport was under construction to lengthen the runway, so I couldn't fly there. So I had to fly to Kisumu. And then I was going to have to take a taxi from Kisumu to this like little village, and then take a ferry across to Mbita, so another village, and then someone would come get me in the Land Rover and take me there. Then the problem ran into that I was flying into the Kisumu. I was supposed to originally fly into Kisumu the same day that Obama was visiting his grandma. And so I had to go a day early, and there were all this security there preparing for him. So it was very exciting. But yeah, it was fantastic. They set up my tent for me when I got there, and I had previously done field work in northwestern New Mexico, and that's the sandiest place I've ever been. And so when they were setting up my tent, they were just so judgmental of the amount of sand in my tent, and they like swept it out for me. I'm like, I'm so sorry, I tried. Sand just gets everywhere. But yeah, I was only out there for 2 weeks. It was so much fun. I was working with a bunch of undergraduates who'd never talked to a paleobotanist before. My field assistants are just magnificent humans. And it's really funny because like there are some people who are really good at finding bone, right? Like some people just have a sense. They just know where to look. They can just find it. And I am not that person. There are some people who just know where to find fossil leaves. And that was my field assistant, Suleiman. He was so good at it. He's like, oh, have you looked over there? We'd look over there. We'd just find enormous, beautiful leaves. Like they were fantastic. It was so
you
much
have
fun.
to have those people. So oftentimes it isn't the researcher.
Yeah. Oh yeah. They were locals. So the bottom picture, the man wearing the army t-shirt, his name is Mika. This was his land. He had lived there his whole life. Like Mika knew exactly where to find the fossils. And then Suleiman in the red shirt, he lived like at the bottom of Kiahara Hill. So again, like he knew exactly where to look to find leaves. And his son would come and join us and just stare at us as we were digging because like, what are you people doing?
Yeah. Well, let's go over to David and see if we have any audience questions.
Sure. I'll remind our audience to go ahead. If you have questions for Aly, please leave them in the Facebook comments, or like I said before, if you can't, I'm keeping an eye on Gray Fossil Site's Twitter and Instagram accounts. We are a little bit behind schedule due to our technical difficulties, but we can stick around a bit and answer some questions. We do have one from Greg, who asks, do you get non-analog floras which don't fit in the Whitaker biome diagram?
Okay, so that's a good question. All right. I'm so excited. Okay, so the problem is that we are kind of forcing things into these squares. So like basically we're taking something that is a temperature and precipitation and like, well, we would expect it to be this, but we find non-analog communities pretty commonly. We don't tend to find things that don't fit in at all. I think there's we don't tend to find things that don't fit at all. One of the things that does happen in fossils is that we'll go too far to the right on the diagram. Let me pull that up. Right.
And Aly, if you wouldn't mind just again explaining the non-analog.
I would love to. Boom. All right. So one of the things that we run into with modern sites, fossil sites, there we go. Fossil sites is that sometimes they don't fit perfectly in here. The biggest problem that we run into is that sometimes things will be a little bit warmer than we see today. So non-analog would be, there is nothing today that is comparable to this. So one of my favorite things to talk about, I bring this up constantly because I did my master's at ETSU, is talking about the flora at the Gray Fossil Site. The fact that it is an analogous flora. It's the type of, broadly speaking, the type of plants that you would expect to see today. The species are different. The species are not analogous because they're from East Asia, but broadly speaking, you're seeing the same general types of plants, but sometimes you'll find things living together that you would never expect. And that is much more common in older floras. So if you're looking at, so when I, for example, was at the fluorescent fossil beds, just the types of plants that we were finding together weren't communities that are particularly common today. And that's particularly common as you go further back in time, because plants, man, Paleocene leaves are ridiculous because they hadn't quite figured out how to be leaves yet. And so they're just like, we're gonna try every shape. And everything's a sycamore, but that, I digress. So fortunately, working in the Miocene, I'll admit Miocene is kind of easy mode because you have like modern taxa and a pretty modern orientation of the continents. Like even if the groups aren't where you would expect them to be, broadly speaking, it's who you expect to see. So I personally haven't run into that too much, but
Aly, I think you just made a t-shirt. Miocene. Miocene is in the easy mode.
It is though. That's why I love the Miocene, because it's like it is easy mode, but it is just like weird, just slightly weird.
Yeah.
It's so cool. I mean, come on, there are like tapirs in East Tennessee. Like, come on.
All right. Well, if anyone else has any questions, please feel free to ask them. In the meantime, Chris, did you have anything you wanted to ask?
Oh, sure. I've got a question to ask. So, we're talking about leaves almost exclusively, right? So teeth,
Yes.
smooth, thick, skinny. Are there other, and one of the cool things about your work is that you're using these multiple lines of evidence. And so you're looking at, well, the thickness or the number of teeth or things like that. Are there other parts of the plant that also kind of vary with some fidelity to climate parameters? I mean, can you look at like pores in wood or cone size or fruit size or something like that?
All right.
Does that exist?
All right. I love this. Okay. So as far as I know, it's not as well, like, it's not as easy, not as plug and play as you get with like, how many teeth are there with the leaves? I don't know. The other thing about leaves is that they are just far more abundant, right? Plants have so many leaves. I know that people have looked into this. I know that it's something that people have considered, like, looking into what are the flowers telling us? Because you can get some sort of idea of what was going on by like looking at the flowers or looking at the fruits, because it's going to tell you how is it being pollinated, who is dispersing this fruit. But I don't know about fossil wood. I love people who study fossil wood. Like, the wood people are magnificent. It's baffling to me. I know you can do some things with it, but the thing, I'm going to predict it now, and I keep, anytime anyone asks me, I will bring up the same thing. I think the next big thing in paleobotanical, like, environmental reconstructions is really going to be roots. And the reason for that is that leaves have a direct relationship to temperature, because the temperature is going to affect whether or not they can photosynthesize in a variety of ways. It's chemistry, right? It's going to affect the rate of the process and whether or not like they can even do it if they have to drop their leaves. But water, it really is indirect, because the leaves may lose the water, but they're not absorbing the water. That's coming in through the roots. And also by looking at the arrangement, the size, the shape of the roots, you can get a sense of how deep the water table is. You can get a sense of whether or not you have nutrient limitations. If they have mycorrhizal networks, like that's going to tell you something about nitrogen. So I really think that like the next big thing in understanding on kind of like a holistic scale, paleo environments is really going to be considering roots. Roots are so common in the fossil record, and like leaves, it can be kind of taxon independent. You don't necessarily need to know who it is because you're looking at the shape of it. I'm generally just a nerd about shape, so that's kind of how I come at it.
I've got a question for you, Aly.
I'm ready.
You mentioned at one point early on in your presentation, you showed a couple of maps side by side, and you sort of hinted at this concept of the continent of Africa being understudied or underutilized in these sorts of research. And it was interesting you mentioned hominids at the end, because I feel like just sort of in passing, my experience is that most of the time when I hear about African fossil work, it's often paleoanthropology, it's also ancient human stuff. So in your view, from what you know, to what extent is Africa as a continent still under-researched in the kind of things that you're doing, and to what extent is that improving? Okay,
so the thing about it is always going to come down to funding, because it's not that people in the continent don't know what's going on. They definitely do. And I spent a good amount of time in the East Africa herbarium when I was at the NMK. So that's the thing. This data is out there, right? Like this information exists. It's getting funding to people who can do something with it. And that was one of the things that I found so frustrating. So when I was working on this project, and I understand it a little bit now being a collections manager, but I was told repeatedly that digitization was research driven. It was project driven. They were digitizing the things that people were working on. And right now, the new hotness in paleobotany is South America. And there's nothing wrong with that. That's super cool. And that's why we have data from South America in this project, is because there's so many people there working on it. But really, it's just, there are plenty of people in Africa, in Kenya specifically. I know people who are looking at this sort of stuff. It's just getting funding, getting these resources so people outside of the continent can know about it. One of the things that happened when I was at the NMK was that Google, Amazon, I don't know, somebody like that, gave a whole bunch of money to the NMK, the National Museums of Kenya, to digitize their collections. And they started with archaeology, and then they went from there. But they were doing 3D scanning, and it was amazing in order to again, share this with more people. Because I don't think it's that, I think there's a danger in saying like, oh, no one knows this. No, people know this. Just right now, you have to have the money to either like fly to Africa to get there, and like the money, I think, is the biggest obstacle. And the other problem is I tried to get multiple grants to fund this project, and no one wanted to do it. Like, which is fascinating, because when I would talk to other paleobotanists, they're like, this is so important. Someone needs to do this. I'm like, I would love to be that someone, but then I could not get funding for it. I tried to apply to do a postdoc doing this, and no one wanted to fund it. So I think that this is like, there is gold here. Like, this is an untapped resource. There's so much that I think we're going to learn about these relationships by looking into tropical Africa. And other parts too. Like, fundamentally, Africa has some of like the absolute coolest biomes. The temperate regions of South Africa, the Fynbos, phenomenal, such weird plants. They're like Dr. Seuss plants, and they just show another way of being temperate that is not represented if you look in other parts of the world. So basically, give money to people to do research in Africa. Give money to Africans to do research in Africa.
That's great. Let's do one more audience question. And then, well, okay, we have two, and we can keep them quick.
Okay.
Because we are getting towards time, and then we'll turn it over to Blaine for our last question. Dexem asks, do we see stomatal remnants in fossil plants?
Yes, but I didn't get to. So yes, that's one of the cool things that you can do. Cuticle will be preserved. So cuticle is like the skin layer on top of a plant, and you can process it in HF, which is a hydrofluoric acid. It's a terrifying process, but you can use that to look at it under the microscope. So one of my colleagues during my PhD used that. He was studying that in early Paleocene, so like 65 million year old leaves from New Mexico. Unfortunately, my preservation didn't allow that. Last thing, it was really cool, but messed up. There was so much calcium carbonate in the soil on Rasinga that calcium carbonate, so calcite, replaced my leaves in the impressions. So I would have these films of calcium carbonate, which is really cool, but it's so shiny. It was so hard to photograph.
I should mention, since we asked Dexem's question, that they also commented very early on when we were getting started to say, "Hi, Aly. I always listen to you in the Common Descent podcast. Let's go plants." So thanks, Dexem, for listening to the podcast.
Thank you so much.
And then our last audience question. This is from Ethan, our friend Ethan, who has been on Paleo Talks as well, who has asked what I assume is an inflammatory question.
Oh no.
Which is, what is better, leaves or paleosols?
Oh, okay. We're best friends. You can't do that to us. All right. Like, seriously though, the best this is a great question, Ethan. The best paleo environmental data you're going to get is if you have both leaves and paleosols, because plants have a direct relationship with temperature, but an indirect relationship with precipitation. Paleosols have an indirect relationship with temperature and a direct relationship with precipitation. So if you have both, you can take the best of both worlds and like get temperature, get precipitation, get seasonality. Like, no, you are not gonna break up this power couple. Like, you need both. I will hype paleosols all day.
So take that, Ethan. Take that, and how dare you?
Yeah.
All right. Well, thank you to everyone who has asked questions in our comments. Blaine, do you have anything you want to wrap us up with?
Aly, I just want to say, if you could answer your questions with a little more enthusiasm, we would appreciate it.
I don't think I could do that.
I love it. One final question, and this is circling back to the gray fossil site. If you've been here, you've worked on the plants, if we could find something here that would help improve the story that we're building about the site and about the plants, what could we find in the plant record? What should we be looking for?
Okay, I know what you do have. I don't know.
Do we have enough leaves for you?
I've never actually looked at your leaves.
Okay, well, we don't have very many leaves.
I was gonna say, I need at least 15.
Okay.
I don't know. Like I said, I'm really fascinated because like y'all have seeds and fruits, and that is very useful, especially when telling the story of the like how the disjunct populations in East Africa, or not East Africa, excuse me, East Asia and Eastern North America. I don't know. Like, if y'all could find like a stump. You know what I mean? Like something kind of in situ, right? Like something that could give you a sense of the actual shape of like the branches, that sort of thing. I don't know. Like obviously leaves, but that's not the kind of place where you're really going to find leaves. I don't know.
Well, actually, there is a place on the site that produces leaves. We're just not currently digging there.
Well, why not?
Well, that's the answer we wanted.
Go work on the leaves.
Okay.
That's what you need to do. Okay, cool. That was the answer you're looking for. I will
Yeah.
give you that justification. Go work on the leaves.
All right, we will do that. Well, thank you again for joining us. It's been a real pleasure, and just to see you again has been fantastic.
This has been great. Thank you.
Stick around for a little bit afterwards if you can, Aly.
I
Thanks,
will.
everybody.
Thank you to everyone for joining us today. Thanks, of course, to Aly for being our guest. Thanks to everyone for asking questions. Keep your ears and eyes out. We will have more Paleo Talks coming up in the future. We're now on a every other week schedule. So, and then I think that for anyone out there who is watching that is local to us, we should announce that we have an event coming up on Tuesday. Our traditional fossil and artifact ID night is happening Tuesday late afternoon, starting at 4. You can find posts about it on our social media, the Gray Fossil Site social media pages, Facebook and Twitter and Instagram. If you are local and you have some cool stuff you want our experts to identify, you can bring it by the museum on Tuesday. Other than that,
Wait, David.
oh, go ahead.
One final, if you want to read Corey's there, that's a final compliment right here at the end.
Corey said, well, I guess we should ask, Aly, do you teach?
Not anymore, but I do a lot of outreach.
Corey says you'd be great for teaching. And
Yeah.
yes, I'm sure you are and have been.
Yeah.
I'm award winning.
This is what Aly is a regular on the podcast that our friend Will and I do, the Common Descent podcast, because you just bring a lot of enthusiasm, and we bring you on because you do what we can't do, which is, as you like to say, speak for the trees.
You need one plant person. It took y'all way too long to grow plants.
Aly is our friendly neighborhood paleobotanist that we have on the podcast.
Thank you again. Bye-bye.